Thank you. My name is Maaike. I'm a postdoc in Lucas's lab, and I have a background in experimental health psychology, but now my focus is on mind-body, brain-body, gut-brain interactions and the role in persistent somatic symptoms. And today I want to take the opportunity to show you some of our results of a large European project that we were working on, the discovery project. I will explain a bit more in detail. And particularly on the neuro-endocrine stress response in irritable bowel syndrome. So you probably all already heard about irritable bowel syndrome, or IBS. It's quite prevalent. Everyone knows someone with IBS, I think. But for those who don't know, it's characterized by abdominal pain and altered bowel habits, like constipation and diarrhea. There's no singular identified cause of IBS, and it's considered a disorder of gut-brain interaction, with the idea that several mechanisms along the gut-brain axis are dysfunctional, like dysbiosis of the gut microbiome, increased intestinal permeability, and of course also central mechanisms that I will talk about. What's interesting about IBS is that it's often comorbid with psychiatric disorders such as depression and anxiety, but also with other functional somatic disorders such as chronic fatigue syndrome and fibromyalgia. And we think that stress and the stress response system play a large role in IBS. Why do we think this? For several reasons. First of all, we know that chronic stress and early life stress are risk factors for the development of IBS. We also know that if we look at an IBS patient, they often report that stress exacerbates their symptoms. There's also studies on this. This is actually a study that was carried out in Maastricht. They have developed like an experience sampling app specifically for IBS, in which people get like 10 beeps a day on their cell phone, asking about abdominal symptoms, asking about mood. And we see that there is quite some covariation between patients' reported stress and their abdominal pain. It's not perfect, of course, but we see that acute stress exacerbates symptoms in daily life in IBS patients. Third, of course, stress response systems such as the HPA axis and the autonomic nervous system play a large role in gut-brain and brain-gut communication, as Lucas has already nicely shown. And there's also substantial, not always very consistent, but there is some evidence for HPA axis and ANS dysfunction in IBS. It's not always consistent. For instance, with HPA axis, you sometimes find hyper-responsiveness, sometimes you find hypo-responsiveness in IBS, sometimes you find no differences at all. With ANS dysfunction, I think it's quite consistent that we see lower HRV in IBS patients compared to controls. So there is at least some evidence. As I told you a couple of slides ago, there are some disorders that are often comorbid with IBS, such as anxiety, depression, fatigue, chronic pain. And we think that one of the reasons that they are so often comorbid with IBS is that they also share this underlying mechanism of dysfunctions along the gut-brain axis, including difficulties with stress regulation. And we also know that in patients with IBS, actually those with higher comorbidities have lower quality of life, they're harder to treat, and they pose a larger socioeconomic burden. And this is where the discovery project I was talking about comes into the picture. The goal of this project was actually to investigate whether higher levels of comorbidities reflect higher disturbances along the gut-brain axis. So what we did, or what the consortium was about, is to recruit a large group of IBS patients and healthy controls with and without these comorbidities, and look at several mechanisms along the gut-brain axis. And I think in the upcoming year, a lot of these studies will be coming out, will be published. But today I specifically want to talk about the work packages that Lucas and I were working on the stress response system in IBS and the relationship with comorbidities. So what did we specifically want to do in this work package, or what will I present today at least, is we want to investigate self-reported responses to acute stress, cortisol responses to acute stress, to look at HPA axis function, and neural responses to acute stress in IBS. And the latter is actually something that, as far as I know, has not been done in IBS, at least not separately from also giving painful stimuli, for instance, in the rectum. So we wanted to look at purely stress and then see what the neural responses were. And we first, of course, had the goal to compare these things between IBS patients and healthy controls. And in the second goal, we wanted to look at the role of comorbidities. And we did this in two complementary ways. First of all, we compared patients with and without comorbidities, looking at it really categorically, checking whether patients fulfill the criteria for anxiety, depression, fibromyalgia, or chronic fatigue syndrome. But then complementary to that, we also looked at it in a bit more of a continuous way and looking at comorbidity severity outside of these categorical borders. How did we do this? So we recruited a bunch of IBS patients and IBS diagnosis was confirmed using the Rome 4 criteria. We did the mini international neuropsychiatric interview with all those patients to see whether they fulfilled criteria for an anxiety disorder or for depression. We also checked the criteria for fibromyalgia and chronic fatigue syndrome. And patients filled out a couple of questionnaires, among which the PHQ-9 measuring severity of depressive symptoms, GD7 measuring anxiety symptoms, the multidimensional fatigue inventory measuring fatigue, and the fibromyalgia impact questionnaire measuring the impacts of widespread pain. And what we did then is we wanted to have one singular score. So I did a principal component analysis on this, which showed that actually most of these questionnaires loaded onto one single factor, explaining, I think, about 70% of the variance. So we just derived the factor scores for that factor from each patient or each participant. And we used that as a measure of comorbidity severity throughout the studies. As I said, we took measures along the whole gut-brain axis, but I will only explain what is relevant for us or for me today. So a subset of the complete discovery sample underwent an imaging task, the Montreal imaging stress task. I will explain in the next slide, and also the Maastricht acute stress test, again developed here in Maastricht, to measure HPA responses to stress. And this was, of course, not in the scanner, but just in the lab. So the Montreal imaging stress task is a task that we use often in our lab to induce stress in the scanner. It consists of a rest condition, control condition, and stress condition, which are alternated. This is what participants see. During the rest condition, they don't have to do anything. During the controlled condition, they are asked to solve some math problems. And they have this dial thingy here. They have a response box and they have to indicate the right answer. It's always the number between 0 and 9. And in the control condition, we tell them, this is like the practice phase. You can do it in your own pace. Just take it easy. And during the stress condition, they actually have like a time limit that they have to solve the math equation. And we also tell them that they're playing against someone. So actually, before they enter the scanner, we introduce them to their opponent, which is a fake opponent. It's always like a master student of someone from the lab. They don't know that. They also don't think too much about it, but it works. And they can actually see the performance of the opponent. And of course, it's made it this way that the opponent is always outperforming them. And if they get a bit too good at the math problems, it gets a bit more difficult. And if they're getting too bad, it gets a bit more easy. That way they always have a glimmer of hope, but they never win. And in between the trials, we actually give them negative feedback, with the latest one being, I actually don't know if we will be able to use this data because you're really not performing well, blah, blah, blah. And they also give stress, negative ratings in between. The Maastricht acute stress test, Lucas already explained a bit. It's an alternation between hand immersion in ice cold water. We use 6 degrees Celsius, which is a bit warmer than what is typically used, but yeah, these are chronic pain patients, so didn't want to torture them too much, and it still works. Alternated with mental arithmetic, and we take saliva samples to measure salivary cortisol before, after, and up until 55 minutes after the end of the MAST. And we also ask people to rate their subjective stress levels and pain and unpleasantness before the MAST, during the instructions, during the MAST, and immediately after the MAST. These are the patients and healthy controls that were recruited for this purpose. They were recruited at KU Leuven, at the University of Gothenburg in Sweden, and here in Maastricht. There were 177 IBS patients, of which 73 did not have any comorbidities, and 104 fulfilled the criteria for at least one of the of depression, anxiety, fibromyalgia, or chronic fatigue syndrome. And if you look at the comorbidity severity score that we created with like these PCA-derived factor scores, you can see that, of course, people with comorbidities have a higher comorbidity severity or a higher comorbidity burden, but it's still quite overlapping. So that's why I think it's very important to look at it both ways, because it's not simply the same thing. Of course, not everyone participated in everything. Not everyone wants to go in the scanner and solve math problems. But these were like the final sample sizes for the MIST and the MAST respectively. Okay, now up to the exciting stuff, the results. First of all, we looked at the self-reported responses to stress. And if we look at the patient versus healthy control comparison, we can see that IBS patients report more stress, but this difference is already present at the intercept, so before starting the MAST, and there was no times group interaction, meaning that the way they react to the stress is not different between patients and controls. It's just they already have more stress overall, nothing to do with the task. And the same was actually found when we compare patients with and without comorbidities. People or patients with comorbidities, they have higher stress. And the same was also found when looking at it continuously with comorbidity severity. So IBS patients experience more stress than healthy controls, but this has nothing to do with the task. And it's especially pronounced in patients with comorbidities and higher comorbidity severity. When looking at the cortisol responses to stress, unfortunately, we don't have the results yet for the healthy controls, so I cannot present you any data on the case control comparison, but we have the data of the patients. So if you look at the comorbidity status, and especially it's very pronounced in a comorbidity severity analysis, that patients who have higher comorbidity burden, they actually have a more exaggerated cortisol response to stress, which is kind of what we expected, and maybe also kind of not what we expected, because like if you look at cortisol literature in depression, anxiety, chronic fatigue syndrome, it's also sometimes hyper-responsiveness, sometimes hypo-responsiveness, but yeah, it's an interesting response. When looking at the neural responses to stress, I first want to explain a little bit how we investigated it. So the basis of all our analyses was actually this contrast. We compared brain activation during the control condition with brain activation during the stress condition. And this is on average what that looked like, meaning that the task worked really well. I think you have a really large difference between the two conditions. And we do this so that you really have the brain during stress, subtracting all the effects of like just the math, doing the math and moving with the hands and all the visual stimulation and so on. Okay, so we extracted this for each participant, and then we did a number of things with this. I'm just gonna tell you a couple. First of all, we of course just compared this contrast in patients versus healthy control, in comorbid versus non-comorbid patients, and looked at the association with the comorbidity severity score. Then using support vector machine algorithms, we tried whether we could predict whether a patient was a patient or a healthy control, whether a patient had a comorbidity or not, and patient's comorbidity severity score based on these bold responses to stress. And third, we compared the neural pain signature activation to stress in patients versus healthy controls and comorbid versus non-comorbid patients. And I will come back to this a bit later, what this actually means. So looking at comparisons between patients and healthy controls, we can actually see that patients react a bit differently to stress in the brain. So the regions that are warm colored, yellow colored, are regions that have higher activation in stress in patients versus healthy control, and lower activation in stress versus healthy controls are the blue regions. So we see that there is an altered neural response to stress in IBS patients versus healthy controls, which is, I think, we will be the first to show that. But we did not find any significant differences in brain activation to stress in comorbid versus non-comorbid patients, neither with the comorbidity severity score. There was like no relationship whatsoever. Then when we turn it around and we feed this brain response into an SVM algorithm, we actually found that based on the stress response, or to the based on the neural responses to stress, especially based on the regions here in yellow, we can quite accurately predict whether someone is an IBS patient or a healthy control. But this was not the case whatsoever for the comorbidity status or the comorbidity severity score. So I think this is also very interesting. Stress, IBS, and the neural pain signature. First, let me explain to you what the neural pain signature is. This is actually a pattern of brain activation that is predictive of pain ratings during experimentally induced pain, with some regions having positive weights, meaning that higher activation in this region is predictive of higher pain ratings, and some regions have a negative weights, meaning that activity in these regions is predictive of lower pain ratings during experimentally induced pain. So we wanted to investigate the difference between IBS patients and healthy controls, and comorbid versus non-comorbid patients in activation of the neural pain signature during stress. And what we see actually is that in healthy controls, the signature with the negative weights is positively activated. What does this mean? During stress, healthy controls engage brain regions that actually down-regulate pain, which is, from an evolutionary perspective, a smart thing to do. But IBS patients fail to do the same thing, which is quite interesting, I think. Again, we found no differences between comorbid patients and non-comorbid patients. So in the brain, it's all about the case control differences. Okay, I'm already through my slides. So some general conclusions. IBS patients report higher stress levels than healthy controls, but this has nothing to do with the task, apparently. And this effect also appears primarily driven by those with a high comorbidity burden. And this is also corroborated by the elevated cortisol responses to psychosocial stress in patients with higher comorbidity levels. But the neural responses to stress differentiate IBS patients from controls, but cannot differentiate patients with and without comorbidities, or patients with varying comorbidity levels. And IBS patients fail to activate pain inhibitory brain regions under stress. I also want to thank the lab. With the same outdated picture, I added a second picture to have some of the people that are not in the first picture, but it's still not complete. I want to thank especially the PhD students, Lichin and Inse, who've been working on this, and Leon Bergwoods, who's also been involved in the discovery project. And of course, the other people who collected data for this work package in Maastricht and in Gothenburg. Thank you.
Thank you very much. Yes, question here.
Thanks very much for the very clear presentation. I have two questions, and that is, I'm also working a lot with patients. So one of the questions that always pops up is it meaningful to group? So while I see the grouping in healthy controls in IBS patients, if you want to call them patients, the differentiation between those with and without comorbidity is a quintessential question for me, because don't you think that those who may not have had a comorbidity may
Develop it later on
along the way? And so would a continuous type of analysis make more sense to you, or is that something that you have entertained?
Well, we did it.
Oh, maybe then I overlooked it. Sorry.
Yeah. So
because I thought you didn't find differences between the two groups.
no, but here I know. Yeah, you see 3 different lines, but this was not a classification.
Okay.
These are estimates based on someone with, for instance, a low comorbidity severity, meaning like 1 standard deviation below the mean, and but in the analysis, this was done continuously. This is for graphical purpose.
Just a bit of background. I mean, we did this exactly because of the reasons that you were mentioning. It was not really foreseen in the project, but of course, this is a horizon project, so you always have to address every single stupid word in the call text. But it was a call on comorbidities.
Yeah. Okay.
So we had to do the categorical approach, but then we ourselves, and this is now going to be adopted across the consortium, took the initiative to say, okay, we want to look at this in a continuous fashion, right? And that's how. I also think so. So
it's statistically more powerful.
But indeed, I mean, this is basically plotting at the average and 1 standard deviation above and below the average of that continuum, but it gives the impression of being categorical, but the right-hand side is not. Exactly as you, I mean, for the reasons that you mentioned.
Yeah. Also, it turned out to be a bit, I think, biased, this categorical approach, because between the centers, there was a lot of differences in how you define it, and when is someone, also the neuropsychiatric interview, from experience, I know that people who don't have a psychology or psychiatry background will be a bit more easy to say, oh, that person is depressed, that person has an anxiety disorder, while we have a bit higher standards to meet that cutoff. So I think, this is a bit more...
Yeah, it's also less biased.
Less biased. Of course, there's differences in languages between the questionnaires, but I think that's less of a problem. With your remark about, they might still develop a comorbidity, I think it's nice to highlight that this is actually also a longitudinal study. So these patients were followed up for 2 years, and we did the mini again, we did the questionnaires again. So it's not a long time span, but at least we will be able to also say something about that.
That's great. Tiny additional question. So I'm a bit, I'm curious to hear why you think in the IBS patients you see increased, if I understand this correctly, increased activity in the caudate and in midline structures. Any ideas what that comes about with?
No, maybe Lucas has a better.
Yeah, it's clear. It's a bilateral caudate, and I mean ventromedial prefrontal cortex. That is something that you very often see with the MISTAT. That is a region that is implicated. Also, the caudate, there are some data on it, but I must say We just recently analyzed those data, and we still need to look into more detail for the interpretation of it, because it's definitely not a region that we primarily anticipate.
I mean, I see these part of a cognitive control system. Right?
Yeah, but
So
not only,
not only, but
it depends on, yeah,
certainly contributing. And I saw in some other breakdown also ACC activate. And so
yeah,
I would say what would be interesting is to indeed see whether if there's a dorsal ventral dimensionality. I mean, this is just a particular breakdown, right? But because in some studies where we controlled for, we looked into emotional control, and there we see a lot of high correlations with anxiety and depression. So I'm just trying to speculate here, but I was a bit intrigued by this. Yeah.
Yeah.
Yeah, it's
Look
really
into
there.
it for
Also in
sure.
the SPM, you see
Yeah,
that it
yeah.
discriminates. So
Yeah.
it's a consistent. Yeah, you get a call
Yeah.
again. So yeah, it's a good point.
Do you also have resting state data from the patient?
Yes, we do, but we have not analyzed it yet.
Because
It's,
my
next
question-
on our to-do list.
Yeah, that's next one.
Because my question would be, if you have mapped these regions that you highlight now relative to the gastric brain network that is usually identified from Rebolo and colleagues, because some of these regions actually are part of the gastric network, so it may be that, okay, now are somewhat task-driven, so modulated in these patients, but actually even at rest, there is an either hyper or hypo representation of these regions, right? Because then they sort of increase the sensitivity to gastric symptoms in patients.
And then of course, IBS is a large bowel disorder. It's not a gastric disorder. I mean,
no, true, but they do gastric to
to the point that I think Valent made yesterday. I mean, studying the stomach in IBS, how meaningful is that?
Yeah,
I'm not entirely sure. But definitely the resting state data, I mean, of course, we will interpret in that way, yeah, but it's something that still needs to be done.
But I mean, I'm thinking this could really be something about developing anxieties, right? Because if you have that symptom, it makes you think about what you eat. I have two cases in my family, so I see exactly what that has done in terms of behavioral change towards food.
Absolutely. Yeah.
And whether or not that's sensible or meaningful doesn't really matter, right? Because it's like, I know it will lead to something.
It's certainly something that is also described a lot in the literature, this concept of gastrointestinal specific anxiety, where people really develop very specific fears towards symptom inducing contexts or foods or things like that. I mean,
which we unfortunately did not measure in the discovery.
I know, which is totally a mistake, because the most relevant psychological thing to measure is this GI specific anxiety. My limited amount of clinical work, which is half a day a week as a psychiatrist, is in patients, functional GI patients with comorbidities, and this is something that we see all the time, and that we treat typically with exposure-based CBT. We treat it like an anxiety disorder. Before the whole paper on the VSI and GI specific anxiety came out, I used to call it gastrointestinal agoraphobia, because it shares a lot of the features of agoraphobia in the context of panic disorder. So it's a very good point, that this is a really important part, and you get sometimes very high levels of this GI specific anxiety in people who don't have any anxiety disorder comorbidity. It's something very specific, which makes a lot of sense. Of course, people experience that, okay, if I go out and eat, I get symptoms and things like that, so they develop. It's just, I mean, it's elemental learning psychology. So yes, so it would be very nice to be able to interpret things in that context, but for a reason that I don't remember, it was not included in the consortium, that questionnaire, which is, I mean, I'm sure I complained about it at some point, but
I was not involved yet.
They do. No, that's true. It was before your time, but yeah, it's a good point, but unfortunately, we don't have the data.
Yeah, because you were mentioning it was a longitudinal study, and
Yes.
that was like 2 years, right? So
Yeah.
I was wondering whether patients were also receiving the treatment during.
Treatment as usual.
Okay, so it was
Mhm.
both like medical and psychological treatment. Or...
It will depend on the center, I think, also where they were recruited.
No, because
I think primarily medical, but
Right,
we can get more
medical, yeah.
light on that. Working in
Yeah, it
clinic.
was just not practically feasible to like standardize treatment for
No,
such
yeah,
a long
yeah,
time,
yeah.
of course. But it's a good point.
No, because I was thinking that maybe also the fact that you didn't find differences between patients with and without comorbid disorders maybe could have also been to the fact that they were also receiving treatment during that period.
For their
for their
comorbidity,
anxiety or depression.
Yeah. Yeah, but, yeah, we can look into medication and stuff, but I don't think we will have like the statistical power to really do something with that.
Yeah.
But we do have some information, yes.
You take the last one, Laura?
I was wondering, so the things you measured as comorbidity severity in your patients, did you assess them in your healthy controls as well? To see if they have like it sounds, it's not a comorbidity in that sense, but
Sensitivity.
yeah. Would they look similar to that left group here, or what would you?
It's certainly something to look into, but a large group of the healthy controls we actually borrowed from our chronic fatigue syndrome project. And there we did measure anxiety and depressive symptoms, but not MFI and FIQ. So we could look at depression and anxiety.
How you would found people without any of those symptoms.
Yeah, it's a continuum, of course. Like they're not all gonna be zero, but at least for the healthy controls, we know that they do not have any, like do not meet any of the criteria for those things.
So I do expect the distribution to be similar and
maybe even a bit
little
more
bit
here.
further shifted to the left.
Yeah, with less of a like outliers. Because here, of course, are also people that like you take the mini from, and they just didn't fulfill that one single criterion, so they didn't end up in the comorbidity group, which I don't think there will be for the healthy controls, just because we pre-screen them for that.
But if that applies to like the comorbidity thing, that also applies for healthy controls, not meaning IBS symptoms.
No, we had stricter criteria than that. Like they couldn't have any more than one moderate GI symptom in the past 2 weeks or something. So yeah, we were a bit stricter, and I think also with the psychiatric comorbidities, like if there's a hint of depression and anxiety going on, then or chronic pain or chronic fatigue.
I think it will be a narrower, more leftward shifted distribution.
For sure. Yeah, but we can look into that, at least for depression and anxiety. Okay,
thank you very much again.